paper, I can't remember what it's called. But it's like mesh implant in the stomach of the mouse, and within the liver, they saw an increase in fatty lipids, which you see with aging.
Mm.
I think a lot of the models we use are very related to aging.
Yeah. It's definitely in that way. And then so for the life of me, I couldn't figure. So there's a lot of we do a lot of RNA-seq, and so we've engineered proteins to pretty much do the same thing that epigenetic drugs do, but in a more understandable way. But I believe we've got reduced pleiotropic effects with our engineered chromatin proteins. But it's just striking. It's like we see the same sets of genes getting activated over and over, right?
Yeah.
So the innate immune pathway genes tend to be overrepresented in the genes that get upregulated when we do genetic treatment. Interferon signaling. I think interferon, was it 1 pathway? Like for these transcription, IRF transcription factors. And
you know which? Are you playing cells for ice
human eyes, yeah.
Male or female?
Well, assume female breast cancer.
So interferon is interesting because it's on your X on the portion that has some XISD. I can't remember how to pronounce it.
So I was actually expecting, well, I've been saying this. I was expecting to see a lot of excellent genes come up, but we don't.
Yeah.
So they're mostly on the autosomes.
So I think interferon is an interesting pathway where you see sex differences as well. So seeing the same pattern in male versus female.
Oh yeah.
Or biologic male versus female. It'd be interesting too, because we've seen interferon gamma response or interferon. I don't think it's gamma. I think it is type 1 in our mouse model.
Oh, okay.
In our single cell sequencing of our T cell.
Oh, right.
We do everything with female mice.
Right. But that's what you see an increase in that.
We see a specific type 1 interferon response.
Okay.
Through our fibrosis. We haven't done anything yet.
Yeah.
That's cool.
Yeah. Or moderator.
It is recording, so
okay.
In theory, someone has been responsible for that.
Yeah, are any of our Zoom participants assigned to be the moderator for this breakout session?
Are we muted?
I'm the virtual moderator, but I'm under the impression that there's supposed to be an in-person moderator as well.
Okay. Oh, okay. So I guess we're kind of stuck over there. All right. Well, unless anyone else wants to volunteer to, I guess, record. Actually, let's do it this way. So we can have a moderator and someone to record and report out. So we can split those responsibilities up. I'll volunteer, I'll moderate. Since I had kind of promoted this theme. Anyone want to record and report out?
I'll record.
Okay, awesome. All right, thanks, Rick. Okay, well, so when we were in the sort of the general meeting, we had identified some, sort of larger overarching themes to, I guess, sort of build some more specific research project ideas based on. And the theme for this breakout room is, I guess, sort of developing ways to better understand aging at the cellular level. So synthetic biology tools that can help us to better interrogate the aging process within single cells. And so I emphasize, so that doesn't mean that the system would be limited to single cell systems. So I guess what I mean by that is, you know, processes like chromatin remodeling, cell signaling, you know, even intracellular, you know, signaling cascades. So these specific processes that, you know, where, you know, the literature, right? So studies that have been studies are reporting, you know, sort of tracking these changes in, you know, chromatin signatures and different metabolic states. And it seems like a lot of the work is observational, right, and correlative. And then so the idea here is to come up with creative ways to actually control specific processes in cells to determine causality and maybe even to model aging, right? So if you could actually, you know, induce aging through, I guess, sort of more precise means other than irradiation and poisoning the cells and chemo, right? Can we actually go in and use molecular cell engineering to induce an age state? And then so the importance or the potential impact of doing that would be like, let's say a cell line, right? That you could share with, you know, other labs, and different labs could use for drug screen. So if they're interested in sort of screening compounds to reverse aging, you could use your engineered cell to start with an induced age state and then screen for compounds that would reverse that. And then also sort of one idea that I suggested was, well, you could even sort of determine, you'd have a nice platform system to determine whether some pre-existing drugs for other treatments where, you know, it's suspected that those accelerate aging, like chemotherapy, right? It's a good example. You could use this sort of in vitro cell platform or grow them into organoids or whatever scale you want. Again, induce the age, or sorry, start with a you know, non-age state and then see if that would drive yeah, I guess the application of that would be start with non-age cells, have some synthetic, you know, plug-in in the cell that could very easily report out whether the cells were being aged prematurely. I mean, that's, I guess, another sort of approach to that. Yeah, okay, so that is sort of my overview of the theme for this breakout room. So any questions or maybe even additional suggestions about how we should define this theme?
Oh, that's a good way to start.
Okay, great. Okay, Rick, did you get all that? Okay, all right, great. Okay, so we don't have any specific prompt, which is making this a little bit challenging.
It makes it fun though.
Yeah, it is fun.
So our lab works in senescence as well, aside from immunology. And I think one thing that could be really interesting, and piggybacking off of our lab, doesn't trust senescence-induced radiation to be representative of in vivo. We also do chemo as well.
Right.
If there was a cell line that we could reliably go, this does replicate some version of in vivo senescence, depending on which lab you talk to.
Right.
Injury senescence versus aging senescence are different. Who knows?
Yeah.
If we could make that in a dish, being able to study this is truly a senescent cell in a dish is also just
yeah. So I think so. One, you know, as much as I, you know, have, you know, my fair share of criticisms for this Yang et al. Cell 2023 paper, the ice mice, I think this ice mice example, right, it inspires me to kind of think along those lines. So basically what they did was that, and again, I'm kind of confused about why they chose the double strand break repair activation as like a way to, I mean, there's probably some backstory. So, you know, who was it? Yeah, Sinclair and his group. I mean, he's got, you know, a sort of a trajectory of research dating all the way back to 1999. So I'm pretty sure there's a lot that led up to this. but it's that paper. So I don't, does anybody, first of all, I'm talking about this as if everyone's seen this paper. Anyone not familiar? Yeah, not familiar or hasn't had time to read this paper. Okay, so the reason why I bring it up is that this is almost kind of like akin to like irradiation, but instead of just doing, DNA double-stranded breaks willy-nilly, they chose to do it enzymatically. So they overexpress an enzyme that, and this is incredible, it only has like, I think 20 sites in the entire genome that it will go and cut. And they did all this fancy genome-wide sequencing to show that, yes, this is exactly where it cuts. So they did these, very, sort of sparse, precise cleavages across the genome, and that was their inducer. So I guess sort of what inspires me is that it's like, okay, well, could we do epigenome editing, which is sort of a fancy phrase for, taking a synthetic fusion protein that has a DNA binding domain, and then another domain hanging off of the C terminus usually, that can go and recognize a histone tail, for instance, and then methylate it or acetylate it or remove those marks or phosphorylate. So you can actually deliberately change the epigenetic marks at specific sites in the genome. So that's an established tool. Charlie Gershbaugh, right, is very well known for doing this. And then you can also control DNA methylation. You can methylate and demethylate. So synthetic biologists have been busy developing like this epigenome editing toolbox, and it works, and you can do it in a lot of different cell types. So that would be, so then sort of doing that in the context of aging, I'm not aware, Rick, you've been looking into this. Does anyone use epigenome editing in the context of aging research?
Not that I know.
So that's why, and then so I guess one of the reasons why I volunteered to be in this room is that I was like, oh, well, I didn't think that epigenetic engineering in this setting would warrant its own like big thing, but I could see it as fitting under this because there's lots of other sort of like very specific processes in the cell that you can manipulate. Now, I think a good place to start though, I guess what I'm kind of curious about, and this is where I'm kind of leaning on the aging research expertise. So when you do the correlative research, right, when you use like an actual aged mouse or tissues from an aged person, and you go in and you look at epigenetic marks, metabolites, proteome, whatever, what are sort of like the top, I guess, sort of signature states that people have detected that correlate with aged cells versus young cells? Because that would be, I think that would make a good candidate list for the things that a synthetic biology tool should like be used to induce to see if you could actually like then change the phenotype in a way that seems relevant to aging. So I know there's this epigenetic clock, right, concept, right, where you've got, DNA methylation showing up in specific places. I could see a project where synthetic biologists say, okay, well, it'd be challenging to like hit all of those different sites, especially if it's more than 100. Doing epigenome editing. Actually, I have a solution for that myself, but it's a little selfish plug here, but doing epigenome editing at like all of those sites, right? That could be the goal. Like we want to induce methylation, right? Hypermethylation at these 200 sites to see if you do that, is that sufficient to then trigger a cascade where you see like this aging phenotype, right? And so I'm pretty sure there are other things, right? Other features that aging researchers have, concluded, right? Correlate really tightly with age cells, unless the epigenetic clock. So like telomere shortening, is that?
Telomere attrition is one.
Yeah.
How much does the field know about like genetic pathways that get activated?
So aging is like more newer to me, primary drug delivery and biomaterials. From my understanding of a lot of papers is you kind of pick your favorite pathway and then really focus on that. So that like broad view of like, let me look at everything and then narrow down, I think is actually probably missing.
I was thinking,
anyone can correct me there.
Yeah, like could we do like a similar approach to like refactoring like synthetic biology, refactoring the gene pathways, but doing it with the aging pathways to try to like also figure out how all these genes are connected with each other. Like,
Yeah.
to try to understand it, but then if it actually works, then we would have a model that we could then induce with whatever like external stimuli that we want.
Is that directing us back to like the stem cell proliferation pathways? Like we know, and that kind of goes back with the epigenetic studies as well. If we induce stem cell genes for potency factors, it seems to create younger epigenetic state.
So I'm thinking more in the way that like you have to build it to, or like build to understand, or like to prove our hypotheses about how all these genes are like related to each other.
So I would be concerned about the scope. Oh, so just to recap really quickly. So we're so we just sort of you know, restated the theme of this breakout session. And then so I'm you know, suggesting that we kind of start the discussion by listing sort of molecular, you know, cellular features and pathways that have been implicated as being associated with aging through correlative studies. So if we start with the sort of major conclusions of correlative studies, we can then start to talk about, okay, how do we artificially generate those states to see what to actually test the causal relationship that has with aging in cells. So the refactoring idea, I guess it would seem I'd be a little intimidated by the challenge of the search space.
Yeah,
are we talking about the
that's
whole
why I'm like asking about like, is there like how many genes are we talking about? Like, I don't know if people know that.
I don't think they know that, and I think it's also complicated by like every tissue is also different.
Yeah,
so
so it
like,
have to be like
we
a
know
different
that
model for every.
Even
So
if you're
maybe
looking at
gene-
immune cells, every single tissue with aging changes differently.
Yeah.
And that's looking at mouse, which is our easiest systematic way to do it. But yeah.
Like longitudinal studies, if we look at genes being translated as proteins in the plasma and look at the ones that change with age, it's thousands.
Right. So many genes have been, you know, found in animal models affect the aging, right? So they affect a little bit, you know, so it depends, you know, your standard. If, you know, the lifespan extension is 10% or 20%, most people use 10%. So in worm, it's not that hard, you know, so many genes have been mutations or, you know, activations found affect, you know, that. But in human, not that many genes found, you know, to be, you know, affecting aging is a very positive, you know, replicate. Two genes being identified. One is the FOXO3, the other one is ApoE. You know? So these two, but the others in the GWAS study, but all the other studies, the difference is so small, it's hard to call. But these two, you know, are kind of winners. You
know, ApoE is so important to wound healing. So if you're having a change of HMI, that's
yeah, ApoE4 is also major risk factor for the Alzheimer's, you know, not just the aging. Also, you know, I mean,
is it what direction of expression is associated with aging?
So ApoE, the 3 is kind of a wild type, 2 is good, 4 is bad. You know? So
if it's at high levels.
So it's not, you know, expression level, it's a variation.
Like splicing.
Right. No, it's a genotype.
Risk variant.
Yeah.
Longevity.
So genotype 3 is the wild type. Genotype 2 is kind of, you know, good, you know, with, you know, I don't know, you know, the activity. I don't think, you know, the expression matters that much. It's a genotype, you know, so genotype 4 is a bad.
I think it's like 14% of the population has genotype the risk variant.
4 is about 10, about 10%, you know, and also depend on the race, you know.
What do we do again?
That's related to.
It's a lipid. Affect lipid metabolism, you know, ApoE, you know, but there are many other functions.
Okay.
But yet animals models, you know, many genes have been identified. So that's why I think, you know, synthetic biology would be much easier if we understand, aging mechanism much better. Right? But now it seems, so many things affect the aging.
Yeah, so this is why, I guess that's why I was a little bit inspired to kind of take this approach where it's like, okay, well, what I think a good start for sort of at least, coming up with some ideas of, what kind of synthetic biology tool to build. I mean, it has, this approach, it has its shortcomings, right? But I guess sort of my general strategy was to, basically start with specific features that have been correlated, right, with the age state. And then you could use synthetic biology just to do some initial hypothesis testing, right? So is this a driver? Is overexpression of this, sufficient, right, to induce, a general cellular, maybe senescence, right? Maybe some other feature that is also related to aging, but maybe doesn't completely encapsulate, everything that happens during aging, but at least, sort of testing what that feature's causal role is in cell function. So I think that without a total biological understanding of what an age cell is, you can at least start there and start testing some ideas with synthetic biology.
Right.
And then so the nice thing about, synthetic biology, at least sort of the more sophisticated expression systems, is that you can tune the magnitude of expression, you can tune the timing, you can, create feedback loops so that you can do like pulsed expression of things over time. You can link the expression of something to some other change or input. And so I think that you would then be able to potentially sort of get a response. Because I think these experiments where you just simply, you introduce something to the cell, overexpress it, and then you don't see anything, it's like, okay, well, was it under the right conditions? Was it expressed in the right pattern? And so those types of experiments are very limited. So where you, but when you start sort of introducing these more sophisticated, protein expression systems, I think that presents a greater opportunity to maybe observe something that you would have missed. With other approaches.
Right. So I think, many things, we want to do depend on, the mechanism we understand.
Right.
It's not very, if, what we found is right, I think maybe, there is something, we can do. So for example, in the aging study, right, we showed in young, organism, the hydrochromatin very tight. Then in the, aging organism, the hydrochromatin become more open, right? Then you have this retrotransposon that activated, I mean, there are many studies on that. So it seems, pretty good, study. But if that's true, right, can we use synthetic biology to put some kind of, things to hold the hydrochromatin structure together, right? So then, then they don't open up. Then retrotransposon don't get activated.
So here's another thing, observation that I've been intrigued with. So given that sort of that trend, right? So it seems like, okay, more robust heterochromatin is good, right? For more youthful. Cancer cells, a lot of breast cancer in particular. Will amplify a chromatin modifier called EZH2. EZH2 methylates histone 3 at lysine 27, which is associated with silencing. So it's like this sort of increase in building up silencing chromatin, sort of like pushes the cell towards a state that's more associated with like more proliferation. So anyway, that's why I kind of, was curious about, whether anyone was, thought it would be important to kind of follow these parallels, because if you're going to develop a rejuvenation therapy, I think based on what I've learned, you kind of have to watch out for inducing a state that kind of looks like cancer. But to that point, so that makes me really excited because the types of fusion proteins that we build. So we haven't tried this exact trick yet, but this would be something I would propose where, okay, so let's say that the age state has lost some protein-protein interactions that help to compact the chromatin, but maybe the underlying methylation is there. maybe some underlying histone marks are there,
Right.
still, sufficient to act as ligands or signals to then scaffold the chromatin. So maybe the scaffolding is messed up. And then so I would build a fusion protein that would recognize the residual chromatin marks. And then have an extra domain to sort of enhance the scaffolding, overexpress that. So that way you're not doing a DNA specific targeting. It's based on the chromatin mark. So we focus a lot on like this chromatin reader activity. And that's sort of one of the most prominent tools in our tool set for building fusion proteins. We've successfully, so in my intro talk, I didn't have a chance to go into details, but basically the way that protein works is that it was meant to re-express tumor suppressors. So we had a domain that recognizes H3K27 trimethyl, assuming that is enriched at genes that we want to reactivate again. So we sense our activator after that mark, and then we're able to boost the expression of those genes that have been repressed. So I think we could design a protein to try to do the same thing for H3K9 and
if you know,
because the H3K9 has a very well studied, like the chromodomain, is very specific for that mark. So we could use that as a reader and then send So there's, I think, in terms of sort of restoring chromatin domains. So I picked this, and I didn't want that to be the dominant thing, but I think this falls under this general approach where, you identify these very specific features that are interestingly, kind of like showing up, when you compare age to young, and then, okay, well, let's try to artificially manipulate those and see if that's actually a driver for, the older young state in the cell.
So, I mean, they use Yamamaka factors. So they remove c-MYC, so use the fall so they don't get cancer. I think that affect, epigenetic modifications also, probably affect, chromatin structure.
It probably does. So if you do anything that very dramatically affects transcriptional initiation and elongation, there are other chromatin remodeling factors that become triggered to go in and then start doing things to histones at those genes that are being actively transcribed. So I think that maybe I would guess that so I haven't looked into this specifically, but my guess would be that it's a non-specific sort of like effect that influences remodeling of chromatin. But the direct actors, that actually have the enzymatic activity to change the biochemistry of the chromatin are not the Yamanaka factors themselves. And I'd be interested to see what those might be.
You were talking about, the cancer and the aging, right? There are many, similarities in the studies, right? So for example, c-MYC is oncogene. Then, gemcitabine, knock out one copy of c-MYC, because, if you knock out both copies, the mouse would die. So knock out one copy, actually, the mouse has an extended lifespan. Seems c-MYC was also, kind of pro-aging factors. Then, people study naked mole rat. They have, like a 30, 40 years of lifespan compared to 2 to 3 years of mouse. So those naked mole rat also kind of cancer resistant. So
do they? So is anything known about? So I know that there's also, that reminds me of the, what is it, Petro's paradox, with the you'd expect with the scale of an animal, it would have more cancer incidence, but for elephants, that's not the case.
Not the case.
And then also elephants have more copies of P53 than humans. So is there like some sort of, is there like a molecular redundancy that's kind of similar to that? Make it mole rat, or is it a different?
So we have some people study like, the big animals, right? Elephant, a whale, so in general, they develop some kind of specific tumor suppressor mechanism. So one study showed, they have multiple copies of P53. Then their Govanova showed, there are 36 genes, kind of different, so which is more tumor suppressing. Then there are also some studies showing, the promoters, get a change, the promoters of the tumor suppressor genes, so they have a more, active tumor suppressor mechanism. But you're right, those things are needed, right? Because the bigger animals divide, small types, then they have more, DNA damage, they should get cancer, they should die soon.
Right.
It turns out bigger animals, have more years,
Yeah.
than the small animals in general.
Excuse me. Just going back to one of the themes, making a in vitro single cell systems to model aging in the lab conditions. I was wondering, do we have to make a cell that is like homogeneously at the same age, or can we just like start with a heterogeneous a group of cells and screen them using some biomarkers that is highly correlated with aging? To my knowledge, DNA methylation as certain DNA sequences is one example for that.
Right.
And then we can just screen them somehow and select just the group of the cells with a similar age and just go from them. I don't know whether we have the technology to select those cells, but
I mean, you can always do cell sorting if there's a fluorescent quarter.
So
somehow associated with it.
So I know a tool that you get to leverage the cell sorting. I know that so in this chromatin protein engineering space, I was in a professor one, I forget her first name, she's at Purdue University. So she's developed these really cool chromatin mark probes, right? So where they have a reader domain that'll recognize something, and then it's attached to a GFP. So if you have like enrichment, right, of some chromatin mark, you should see like a brighter signal, like accumulation of these dots. That may or may not work, but it depends on how abundant that epigenetic clock feature is. So it's if it's present at very low levels, then it would fall below detection limits for that technology. But that's the one thing I can think of off the top of my head. Yeah, you want to avoid, so yeah, you basically want to avoid having to kill the cells, right? So you don't want to do any nucleic acid probe staining or anything like that. I mean, I think, yeah, so it's, but I think that also, that problem is very important, and it also falls under the purview of the signal amplification sensor group. That, yeah. But that's it's definitely important to know what you're starting with, to have a standard for your non-H cell. Yeah.
Yeah, it would be good to have, it's kind of, marker, right? So of the aging, DNA methylation is one of them, but if we understand, better, right? So senescence, during the senescence, what are the key changes, right? So then identify those genes, so build, synthetic biomarkers, and use that kind of cell as a kind of a system to screen drugs or other things.
Yeah, I wonder if you even need to sort them, because there are systems like there's a progression of aging, right? So if you in a dish already had something that was young, middle, and aged, but you actually have the entire spectrum, and you can figure out a way for each individual cell to see how those change with treatment or whatever you're doing to them. I think that would also have a lot of value in just getting sorting.
I mean, one thing, in the cell, right? So they go in sequence, right? So for example, you can define them, you have the first gene, induce, make it green, right? Then the second gene, make it red. You can actually see the karma change over time. Then that may give you some drug may work on the same, the first gene, another drug work on the second gene. But for all these things, we don't know. For example, we studied senescence for so many years, right? We still don't know the key things, what's the sequence. What we know many gene changes, right? So, but which one are the cause of this senescence? Some maybe, just, kind of a collateral damage, right? Are they important, right? By using synthetic biology, you can connect these genes, have kind of a circuitry of the senescence, right? Which one comes first, which one second. Even successful factors, I don't think, I don't think they are produced at the same time, right? So they,
Yeah.
And also cells factor, different cells produce different cells factors, right? All these things, maybe using synthetic biology technique.
I would jot that down as a general project idea. So developing a synthetic system that could, if I understand you correctly, that could, I guess, make, for instance, express, specific genes in a certain order. Right?
In a certain order.
Yeah.
Then with some kind of senescence, a phenotype readout, right?
Right.
So more precise, then use that kind of synthetic, cell system for drug screening or for some kind of, your idea of, as a sensor or marker,
Right.
What would be a good readout for that?
Right.
Is that like what would we measure in that case?
I mean.
So is SA beta gal, is that affected? And I know I'm familiar with it.
So that was the
first. Is it
first.
affected at the transcriptional level, or is just the protein more active, like post-translationally? Like what is the nature of the increase?
So I assume they are correlated, right? So it could be quite different.
Yeah, I just didn't want to assume. I always assume it's like, more transcription, more protein.
Right,
yeah.
But they always check the protein. I'm not sure they start with the transcription.
Right. Because then that would help you to decide where to put that reporter, right? So it's very important when, sort of making these synthetic biology reporters, where do you think where is it most dynamic, right? Is this being regulated at the transcriptional level, post-transcriptionally, during translation, post-translationally, right? Is that, so there could be just this steady state pool of beta glycidase, and maybe it's I don't know. This is showing my ignorance about beta gal biochemistry, but maybe there's like a post-translational modification that makes that active, that increases the active pool. So so in terms of reporters, it's really important to think about. I always kind of default to making more RNAs, but that's not always the case.
Right. So back to your question of the readout, I mean, so synthesis readout, if we know the pathway, right? So if it's a series of genes, then you can have, the reporters for the, during synthesis, if that gene is activated, you can make a reporter and read out that. You can even have several reporters, right?
Like throughout
the path, then you can see how deep it get into the synthesis.
So I can imagine if you're fortunate and those genes have good sites to, I guess, maybe integrate a fluorescent protein, right? So you could use the bicistronic expression trick. So you basically put like a little P2A tag and then some of your favorite fluorescent protein, make an insertion using CRISPR, hopefully not disrupting the expression of that gene, and then just use the fluorescent tag as a readout. But then I really like the P2A trick because you're not making a fusion. Right. Your fluorescent tag is being expressed as a separate protein but the expression's coupled, but the proteins are not coupled. So I could see that as an actual specific project. System returns in essence both on and off. Oh yeah, would be really important because by definition, senescence often be proliferating. Yeah.
So in the aging study, another manageable system is the mTOR pathway. Right? So mTOR pathway, there is a two complex mTOR1, mTOR2. Activation of one or two or both would cause a very different consequences, right? But it's a protein complex. Maybe it's a manageable system, to try to study just kind of a circuitry of mTOR, right? Then you have drug recognition, which target mTOR. But I don't know how to build that system.
So if it has anything to do with protein interactions, there is a whole suite of molecular biology tools where you can artificially drive protein contacts and pull them apart using drugs or light. There's even like light-based ones, like the I only know I'm old school. I know the protein, like the blue light from plants, right? So it's just you can do controlled dimerization. If you want to force strong dimerization, you can use leucine zippers for that. So that it's there's been a lot of really cool stuff done in yeast to control. Wendell Lim is like really good with this stuff. So he's sort of modified like these, signaling cascades that start at the cell membrane and go down, eventually make their way down in the nucleus. So I think it's the alpha, the mating signaling pathway. He's modified that to control, I guess, the dynamics of kinases that go and assemble at the C5 scaffold. Class, right? Yeah, we talked about this in my civil engineering class. So yeah, so I don't know of any examples of anyone doing that in the context of mTOR. One really popular one for mammalian cells so far has been syn-notch signaling. So synthetic biologists have engineered that to change the signaling dynamics, like the duration of the. So what the initial, ligand finding step, right, stays the same, but what they do is that they sort of rewire the protein interactions that control the magnitude of the signaling once it reaches the nucleus, the timing of it. You can pulse it. There's all sorts of like really cool things you can do by throwing in, little artificial protein interaction domains.
Yeah.
Oh my gosh, that would be awesome to do that with.
And also, for aging, mTOR, it's so important. And and also, it's a complex, I think, a more manageable, do all the kind of things.
I have a question about whether there are really a lot of events that happen in a certain sequence. Because there's so many hallmarks of aging, it looks more like there are many events they do interact, but maybe they initiate in parallel. They don't really need to be a sequence. So it would be great if there's a method to precisely control like multiple events. But is that like possible?
Oh yeah, absolutely.
But is there a limit?
Yeah, so that's the important question, right? So it's like, with the I guess sort of the barrier, right, that synthetic biologists will often hit, that is, just like the sort of baked in feature of biology, is that cells they have very diverse mechanisms, but they're also very efficient in how they use their proteins and like DNA sequences. So they're, depending on the application, so it depends on what your target is, right? So if you're building a system where the DNA, right, is your target for whatever inducer you're designing, the number of, let's say you want to hit like 10 genes with some transcriptional activators. So the size of the recognition site is going to determine how much information is in it. So like if it's only like for instance, a 6 base pair recognition site, you're going to get off target, right? But if you expand out to like 20, that's kind of a sweet spot. And then sort of the combination of ATCs and Gs dictates how unique that site is and how many unique sites you're likely to find. So with so I don't know, that's kind of my really complicated answer to your question about how many things you can control independently because of, just the nature of macromolecules that are used by cells. They each, I guess, sort of like unique target in that scenario. You can only have up to so many before you start seeing overlaps between the molecular composition of target 1 versus 2, 3, and 4. So it just all depends on what you're targeting, right, in that system. I think for DNA, folks have been able to hit, so using CRISPR, right, these CRISPR-based gene regulation systems, I think you can co-express like 10 guide RNAs. So you can control 10 different target genes at the same time. And then there are just other things that sort of like kind of set that limit, like so you actually have to deliver the DNA that encodes all those tRNAs, and then each one needs a promoter. If you put too many promoters at the same time on something, then that's going to be a disaster. So, but you can scale reasonably, but it's not like you can, I don't know if you can like independently control 100 genes, right? At the same time, no one's gotten to that level. Just simply because of the nature of biology. You can only get so much orthogonality within a single cell. Yeah.
It sounds very cool. Just wonder, talk about like precision to like trigger the aging.
Yeah,
But then like I'm just thinking,
but there might not be that many targets that you need to manipulate.
Yeah, like but like let's say you control like a one thing will trigger like senescence.
Right.
But you control multiple things will also get the same physiology. But
Yeah.
like is there actually a difference?
Oh, yeah, I mean, I think that's an important question. There's likely to be, I think. Because if you look at how cells regulate their genes, right, it's like not, especially in early development, there's, you know, this sort of a cascade of gene regulation. Not everything just like comes on at the same time. So, I mean, in general, that's important. You know, the cell makes use of that approach. So I think it's reasonable to, you know, think that timing and order of, you know, control of these different pathways might tell you something, or might give you a specific outcome.
So if we take it as an approach, like you trigger different things separately, and then you compare how the cell react when you trigger them together, is there a way to use that approach to decipher the interaction, I guess? Like although like you see senescence in the end, but if you let's say like RNA-seq the cell, the profile could be like different.
Well, yeah, so you basically have to choose, right, a reasonable output. And I think that's been part of the conversation, right? It's like, okay, well, when we say that we're going to age these cells, how do we know, right? So what's the thing that you measure? And I think my attempt at answering that is that you just have to pick something. And then also when you pick the best thing, right? Maybe it's SA beta gal, you know, maybe it's, I don't know, telomere length or something measurable. But you pick something or a combination of things, and then also just sort of recognize the limitation of that thing as an output. And I think that's the best we can do at this point.
Right. I think one of the example of study, at least for the senescent aspect, I'm just going to read it out on the method. To induce replicated senescence, fibroblasts were passage until their growth ceased completely for at least 2 weeks. They're talking about the in vitro cell culture here. And the senescence was confirmed through various assessments, including the analysis of cell morphology, cell size, and cell expression, and gold standard senescence markers, they call it, along with the gene ontology tests. So can that be like what the examples for?
I think one of the hard things, because this comes up with our lab a lot, is that is very indicative of senescence in a dish. It has little to nothing really for in vivo senescence, because we've seen P16 staining, which is our preferred over beta gal, of small and large cells, but you don't have that like spatial change or that size change and morphologic change when you're looking at a tissue section and looking at P16.
Yeah, that's critical.
So how do you, that also, if you take, I think this is on the Buck Institute website, bulk sequencing of replicated senescence, radiated senescence, and then in vivo senescence, either oncologic or others, like they look nothing.
So that it's like, so the, you know, proliferation pausing in a dish, I mean, it even has its own, what is it, the Hayflick limit? Like this is sort of old observed thing. So to your point, right, I mean, it has its own name, it's, I think
so
that's
guess in
very
addition is
important.
accurate, right? How that translates to in vivo. What in vivo is actually causing senescence? It's like we look at uterine fibroids and there's a massive increase in senescence. Does senescence come first or do they come second?
I think so to really sort of move the needle in the field, right? So we want to do research that's going to take us in the direction we want. I think it's an important point to maybe make a declaration to abandon, right, these cell culture dish specific markers of senescence, and then use the knowledge from, you know, folks who are actually looking at senescence in like tissues and in vivo and use, well, but okay, so here's, I guess the question that I have is that So the markers that are observed in, you know, the 3D environment, could you observe those in addition? Do they go away?
The last one that you mentioned is probably much more what we would use for in vivo. I'm not sure.
Which one?
Oncology?
Yeah, basically. Is it, does it have P16? Is it beta gal? Is it P21I? Is it, did they look at any SAS factors? Argue on what SAS is your preference. Again, we don't agree on them, so.
Well, I'm happy to know that there's a good antibody for P16 because the last time I was a postdoc.
It's debatable.
Okay. Okay.
We get good P16, but we also opal stain and like it's a lot.
Okay.
So, right,
Well,
but
since
the
we were P16,
morphology
that's a
and
good
things like
project.
that, I think that's very true of fibroblasts, but you can have senescent endothelial cells. We have senescent lymphatic cells. I don't know, that in addition with senescent, does that actually represent in vivo senescence?
So then I also wonder, there's many layers of the model systems, right? From the dish cultured ones to organoids that coming up, animal models in vivo, and even human clinical trials. It's like there's many layers of it. What can we learn from the dish can be translated into the more higher level of applications? Because if the phenotypes are so different, like the dish experiments, does that have any significance in the medical field at all?
Oh, we're